now. So our speaker today is Dr. Timothy Vertanian, whose talk today is titled From Gut Microbiome to Multifocal Demyelination in Multiple Sclerosis. Dr. Vertanian is a leading authority on neural regeneration. He's the director of the Judith Jaffe Multiple Sclerosis Center at New York Presbyterian Hospital and Weill Cornell Medicine, and is a professor in the Department of Neurology and Neuroscience at Weill Cornell Medicine. He holds an MD and a PhD in biochemistry and molecular biology from the University of Chicago. Dr. Vertanian has led research teams that have made groundbreaking discoveries in central nervous system remyelination, novel mechanisms of axonal degeneration and regeneration, and mechanisms of immune-mediated injury to myelin and axons in multiple sclerosis. His research interests center on the regeneration of the myelin internode in multiple sclerosis, preventing axonal and neuronal injury, and defining the complicated interactions between the immune and nervous systems. So before I turn it over to him, a few Zoom etiquette reminders. It looks like everybody's muted, which is good. We ask that you stay muted during Dr. Vertanian's talk. Feel free to ask questions using the chat. If you prefer to ask your, after he's concluded his presentation during the Q&A, if you want to ask your question verbally, just use the raise hand feature and we'll call on you to unmute when we get to your question. So Dr. Vertanian, take it away.
Thank you, Andy. I think everyone can see my screen now, hopefully. Thank you for joining today. I was going to give a little bit of background on MS and its treatment, and then talk about the gut microbiome and a potential cause of MS. So MS is a disease that is episodic. That is, patients will develop symptoms. It could be blurred vision, it could be weakness, numbness, difficulty with balance, bladder function. And those symptoms will develop over a period of days, last for a period of weeks, and then resolve. And then maybe a year later, 6 months later, 2 years later, a month later, there will be another attack. And so that's how it gets its name, relapsing remitting MS. The clinical attacks, which are shown here in yellow, are each associated with a new lesion in the brain or spinal cord. And we know that from MRI studies where if we look at the MRI activity, it's actually greater than the clinical activity. And that's because not every place where there is an MRI lesion formed, a new abnormality, a new focus of damage to the nervous system, each one of those doesn't necessarily lead to new symptoms. In fact, only about 1 in 10 of the new MRI lesions will be associated with a new symptom. Large areas of the brain are what we call clinically silent. It's not so obvious when there's an attack. But with each new lesion, there is some permanent injury to the nervous system. And that permanent injury in many patients, untreated, will lead to progressive MS, which is very, very difficult to treat. And as a consequence, the mainstay, the mantra of treating people with MS is to treat as early as possible and as with the highest efficacy medicines as possible while still being safe. Okay, so MS is an illness in which there's a genetic component and an environmental component. We know a lot about the genetics of MS. And we know very little about the environmental causes of MS. But there are some interesting facts to solidify in your mind the genetic contribution. one fact is that if you are an identical twin and one twin has MS, the likelihood that the other twin will develop MS is somewhere between 20 and 30%. So you can have an identical genome, the exact same genetic material, 100%, but you will only have a 30%, 20 or 30% chance of developing MS. This tells us that while MS has a genetic component, that environment dominates risk, that it is something about the environment that is necessary for risk. Some other interesting information about environment. Back in the 70s, the male-female distribution of MS was 63% female, 37% male. In the 1950s, it was 50% male and 50% female. Currently, it's 74% female and 26% male. So the risk, the gender bias risk in MS is changing quite rapidly. This is a very short period of time. So it's changing quite rapidly. Another important fact to know is that the MS has been traditionally thought of as a illness that affects people of Northern European heritage. So people of Northern European heritage, populations that are, have a heavy Scandinavian influence. Well, it turns out that that's actually not the case. If you look in the United States and you adjust for population, so how many white people there are, how many black people there are, how many Hispanics there are, how many Asian Pacific Islanders there are, the frequency of MS is actually higher in the black population, adjusted for numbers compared to the white population. So MS, while the majority of people in the United States are of Northern European heritage who have MS, because that represents the majority of the population, the actual frequency or incidence within these individual populations is as I'm showing you here. Another important aspect environment comes from a number of studies. The best study and the most dramatic study was one in the Faroe Islands. So I've told you that there's a genetic component to MS, and that there is an environmental component to MS. But Andy, I'm realizing I cannot see the chat box now. And so I
can keep an eye on it.
Okay, if you could just monitor it, that would be
Yep.
great. So I told you that there's a genetic component and an environmental component. The genetic component is somewhere between 20 and 30%. The environmental component for risk is somewhere between 70 and 80%. But there have been a number of studies that tell us, like the identical twin studies, that it's not that just that environment contributes to risk, but that environment is necessary for MS to occur. One of the most dramatic studies was in the Faroe Islands, and that's this is just an image of the Faroe Islands. And prior to 1942, in the Faroe Islands, there was no MS, even though the Faroe Islands were genetically Danish, and the highest risk in the world at that time was in Denmark, Iceland, Norway, Finland, all these Scandinavian countries. But in the Faroe Islands, there was no MS. The British occupied the Faroe Islands in 1942, and after that occupation, there was the first of 3 epidemics of MS on the Faroe Islands. So what that tells you is that not only is MS, not only does environment contribute to MS risk, environment is necessary you have to have an environmental trigger for MS to occur. Andy, another thing I'm recognizing is that I'm not sure, are you seeing the people to admit?
Um,
Uh,
I don't think there's anybody showing up. I
uh,
think people are
they've been,
just
they've,
joining.
they've been popping up and I've been admitting them
Oh, that's great. Okay.
because you changed it over to me.
That might be, yeah.
So I'll continue to do that. I just don't want to keep people lagging behind.
I
Um,
appreciate that.
so environment is not only contributory to risk, but it's an environmental signal, environmental trigger is absolutely necessary for MS to occur. So one of the main questions, these are two people who died in accidents. They died at accidental deaths around the same age. They both had MS. This is one person's brain, which looks largely normal except for a few lesions here and there. And this is another person's brain whose MS lesions are dramatically involving vast territories of the brain, and there's a lot of brain atrophy. So why this in one person and this in another person? We don't know the answer to that question, but it is a key question. Why do some people progress and other people don't progress? Why does one person have a relapse at one time and not at another time? Why does one person have 10 relapses a year and another person have one relapse every 5 years? The answers to these questions remain unknown. But it brings us to asking, well, what causes MS? What drives MS? And what all of you have heard throughout, numerous lectures on MS that you've listened to, I'm sure, is that MS is an autoimmune disease. And we certainly know that MS has an inflammatory and immune component. But the question is, what triggers, how does the disease begin? And what role is inflammation playing and where is it playing it in causing injury to the nervous system? So there are two major theories about that. One theory is the inside out theory, and that's that there's some damage to myelin that occurs, maybe due to a virus or a toxin. There's some damage to myelin, and these myelin antigens then get cleared to the lymph nodes, and the foreign substances and resulting in an immune response against myelin. That's the inside out theory. The other theory is the outside in theory. And that's that a person is, living their life, they get an infection by a bacterium or a virus, and that infection, in that infectious organism are molecules that to the immune system look a lot like myelin, or put better, when the immune system raises an immune response against that that antigen, whether it's from a virus or a bacterium, shown here in these little tan pentagons, it is that antigen looks exactly like myelin. So in the outside in theory, the immune response is generated against a pathogen. The immune cells enter the brain through mechanisms that are not well known. And that immune system, which was originally targeted against a virus or a bacterium, now attacks myelin. That's the outside in theory. When we think about treatment, we think about these different compartments, outside and in. And within the central nervous system, there's the myelin and the axons. There are inflammatory cells that normally live within the brain. There's the coverings of the brain. And when we think about the outside the nervous system, we're thinking about lymph nodes. Let me just do that in a little more detail here. So here's the brain. We'll say also the spinal cord and the optic nerves. There's all the structures that live within there. And then when we think of the immune response outside the nervous system, we're thinking about the function of the immune system within lymph nodes, within blood vessels, within bone marrow, that lead to these, that not only the activation of these cells, but early on the production of these immune cells. So just shown here in a little more graphic detail, so we can talk about our current therapies, which many of you who have MS are on or should be on one of these agents. So here is inside the nervous system, and here is outside the nervous system. Here is the barrier between the nervous system and the blood. And so these are just cells that line blood vessels within the brain, spinal cord, and optic nerves. So let's start from outside and work our way in. Not that that's necessarily the right direction, but just as a way to get through all of this information. So if we think of the immune response beginning in lymphoid tissue, in immune tissue, then we have the cells of the immune system, T cells and B cells that regulate immunity, make antibodies, make molecules that can cause injury, and all of that happens within what are called secondary lymphoid tissue, such as lymph nodes. So one drug or class of drugs that we have are S1P antagonists. You don't need to remember that at all. But what these drugs do, Gilenya is, for instance, one of these drugs. What these drugs do is that they target a signal within the lymph tissue that prevents lymphocytes from exiting a lymph node. So normally what your immune system does is it congregates, socializes in lymph nodes. That's where the immune system gets together to see if there is an infection or something that they should be fighting. And so that's where they talk to one another. One of the places they talk to one another. So they'll enter a lymph node to, begin this communication with other immune cells. But these S1P drugs, such as Gilenya, will prevent these cells from exiting the lymph tissue. They can't exit anymore, so they're just stuck there, trapped really, and then they just die. So that's one mechanism by which our drugs work. Another mechanism is shown with these drugs, cladribine, teriflunomide. These are drugs that relatively selectively target the ability of lymphocytes, T cells, to divide by targeting their the molecules that are involved in making DNA. So these drugs prevent the proliferation or the replication of these cells, so that they lower the numbers of immune cells and they prevent the activated ones that potentially could cause damage. They prevent them from dividing. Of drugs are what are called immune depleting drugs. These are typically monoclonal antibodies, alemtuzumab, rituximab, a drug called ocrelizumab, which is like rituximab, or ofatumumab, which is like rituximab. These cells are depleting. So these are antibodies given intravenously. They find their target cell, whether it's a T cell or B cell, they bind to it and they kill it so that those cells are depleted. And so the people who are on those drugs live without those cells for a period of time. And you would think that this would cause great problems in terms of infections, and surprisingly, it doesn't. While there is an increased risk of infections on all of these drugs, it doesn't result in a major problem in most people. A completely different class of drug is called, of which there's really only one currently, are drugs that prevent the interaction of these lymphocytes, whether they're T cells or B cells or even these cells, macrophages. It prevents them from interacting with the blood vessel. So these are called anti-trafficking drugs, and the major one is natalizumab, and that drug targets a molecule on the surface of lymphocytes, and it literally prevents it from communicating with the blood vessel cells that it would normally have to communicate with to get through. And so it prevents all of these immune cells from entering the nervous system. It's like creating a wall. So those aren't all the MS drugs, but those are the majority of the drugs and how they function. There are other drugs, and we don't know exactly how they work in MS. Okay, so let me just check the time here. Actually, I can't see my clock. Andy,
It's 11:23.
Okay, good. So in the last 10 minutes, let me go over this this idea of an environmental agent within your gut microbiome being the initial trigger for an exacerbation or a new lesion to form. One of the things that's been known since the early 1900s is that all MS lesions are centered around a vein, something called a postcapillary venule. So this is shown from early work by a master pathologist, but this is more modern work. Typical MS lesion, and you can see in this MS lesion, it is centered on these veins. Often it'll be centered on one vein, but this is a big lesion and it's centered on multiple veins. So whatever is causing MS lesions to form is probably coming from the blood. So let's go back to environment for a second. So whatever is causing MS is probably coming from the blood. For MS to occur, we've talked about the requirement for an environmental trigger. There has to be something from the environment that triggers MS. So it's coming from the blood. It's coming from the environment, not from genetics. And if we go back to here, we know that it specifically targets blood vessels in the central nervous system because other organs are not involved. So it's specifically targeting brain, spinal cord, optic nerve, blood vessels. So we have been working on a theory that was originally proposed by an Australian physician back in the 80s, that a toxin from a gut bacterium called Clostridium perfringens, a toxin called epsilon toxin, was causative for MS. Now, I'll show you some information why this checks all the boxes for an environmental trigger for MS, which we've been working on very, very diligently over the past 10 years. But let me first tell you a little bit about the toxin. Actually, let me tell you about the organism. The organism, Clostridium perfringens, exists in many places. And there are many different forms of Clostridium perfringens, but two forms, the type B and type D forms, make this toxin. The organism is an anaerobe. That means that it thrives when there's no oxygen. If oxygen is around, it will die, but not quite. It doesn't die. When oxygen is around or the conditions are inhospitable, certain antibiotics, for instance, it will revert to a spore. And a spore is like a capsule that's impenetrable and cannot be damaged. So it is like a time capsule for the organism that prevents it from being eliminated. And when conditions become favorable again, it will grow into a bacteria. Now, the toxin made by this organism is shown here, and it's what's called a pore-forming toxin. This is one molecule, but when it binds to a cell, a target cell, and I'll tell you what those target cells are in a minute, um, it 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 self-assembles into this pore. This was recently solved by a very fancy technique, a very fancy electron microscopy technique. And so this is the pore, the actual pore of this toxin shown at high magnification. So how do we get exposed to this organism? Well, it's an organism that lives in many places. One of the places it lives is in the intestinal tracts, the microbiome of farm animals, sheep, goats, pigs, cows, farm animals, chickens. And farm runoff is a big problem that you might not ever think about, but it is a big problem because it contains all sorts of pathogens. And this farm runoff enters into water systems, it enters into feed, it enters into other crops because this soil is often used, or the fecal matter from these animals is often used as fertilizer. So that's one place. Another place that the organism lives is just in soil. So it's the Clostridium perfringens is found in almost all soil, and the type B and D strains are found occasionally in soil, much more rarely, the ones that make that toxin. The toxin producing organisms have been found in retail food. In Canada, 3.4% of healthy dogs had these toxin producing strains in their feces. So pets haven't. Marine sediment is a common place to find these organisms, and sewage. So what we did was we wanted to disprove our theory. That's what we're supposed to do as scientists, is we're supposed to try to disprove our theory. Our theory was that this organism, toxin-producing organism, was associated with MS. So to test this theory or to disprove this theory, we took a bunch of approaches. This is just one of the approaches. David Sonino, a really bright microbiologist who got his PhD at Cornell University in Ithaca, joined the lab, created a method to isolate bacteria from feces. So this is just a fecal sample that patients or healthy controls donated. It's separated by a centrifuge, ultracentrifugation on a gradient, and the bacteria can be purified. These bacteria are used to isolate DNA and check for the organism, or grown in bacterial culture and the DNA isolated. So using this method, about 30% of people with MS in this one study, this is the only study done to date, this one study, 30% of people with MS had this organism, and 3% of healthy controls. In another study we did, it was 24% of people with MS and 0 healthy controls. But this is an organism that's not supposed to be in humans. And here we have it in 30% of the MS subjects. That's a huge number. And when people do traditional methods, you've heard about metagenomic studies maybe, those methods are not sensitive enough to detect this organism or this toxin. You have to do the method I just described to you. Now the toxin, so one, it's present in people with MS. Two, it specifically binds to brain blood vessels, brain, spinal cord, and optic nerve blood vessels. And when it does so, it causes those blood vessels to open. It makes them permeable. So it opens this barrier, which is normally a very tight barrier between the blood and the brain or spinal cord or optic nerve. So second thing it does is it opens the blood-brain barrier. And then the third thing it does, so I'm just showing this to give you a little bit of orientation. Well, I think this is maybe a little bit too detailed. Well, I'm going to show it to you anyway. This is one of those veins that I've shown you. This is just cut in a different section. And this is brain tissue here. And this is where immune cells can enter the brain if this barrier is damaged. So what Yinghua Ma in my lab did was take animals and immunize them with a myelin antigen, doesn't matter which one. And then he and when you do that, nothing happens. Those animals don't get sick. But if you give those animals this toxin from patients, they develop this illness that resembles MS. And if you look at their spinal cords here, or if you look at their brains shown here, or their corpus callosum shown here, you see lesions, demyelinating lesions throughout the nervous system. And when you look at those lesions in more detail, those lesions, so here's a toxin-induced demyelinating lesion. Those lesions have inflammatory cells exactly like an MS lesion. So that tells us that we've associated this organism with people with MS. The toxin targets blood vessels in the brain, spinal cord, and optic nerve. That's how it specifically targets the central nervous system. And in an animal model, it induces autoimmunity in animals who have been immunized with a myelin antigen. So these are all the people who have done this work. I've mentioned them as we've gone along, and I'll stop sharing my screen here and answer any questions.
Any questions for Dr. Vertanian? Feel free to unmute yourself. I'll start out with one. Are there any websites or other information resources that you particularly recommend for patients and caregivers who want to learn more about this disease?
I think the National MS Society is probably the single best place to go because the information there has been screened by people who are experts in their field, whether it's immunology, neuroscience, neurology, and so I think it's a very reliable site to go to. A lot of other sites that are put up by individuals sometimes have misinformation in them, so I try to steer people towards the National MS Society. Or the other good one is the MSAA, Multiple Sclerosis Association of America, MSAA, NMSS or MSAA.
Thanks. One more last call for any questions from our participants today. Going once, going twice. Is there anything else, Dr. Vertanian, that that wasn't necessarily part of your presentation that you'd like to share with us today?
No, I think that's covered most of it. If people are interested in more information about our work, they can go to my lab website, which is, I'll just type it in the chat box. So they can just go to vertanianlab.com and they'll find it.
All right. Well, thank you very much for taking the time to speak with us today. And I also want to thank all of our participants who joined us today. When Judy is back from vacation, she will post the recording to YouTube. Enjoy the rest of the day. Have a good one, everybody.
Thank you so much, Andy. And thanks for everyone for joining. Take care.